Okay, just a reminder that our next exam is Thursday, April 23rd. That is next Thursday. Everything that we have covered since the last exam will be on it. We last talked about viruses on the last exam, so this exam will include diffusion, osmosis, active transport across the cell membrane. So all these things that we talked about back then on, our first online lecture that I posted for you all. And then also we got into energy. So all the stuff on energy that we have talked about so far, ender and exergonic stuff and enzymes, ATP, oxidation, reduction reactions, all that stuff will be on it, including what's on today. Now, backing up a little bit here, we are coming back to the central metabolic pathways that we're going to be talking about. Getting from C6H12 plus 6O2, and then getting all the way down to 6CO2 and 6 water molecules and a bunch of ATP. So we talked about how that doesn't happen all at once. C6H12O6 does not combine immediately with 6 oxygen molecules to become 6 CO2 and 6 water molecules, O plus ATP over here. To do this, it has to go through the process of glycolysis, and that intermediate step, I can't spell step, the citric acid cycle, I'm abbreviating right now with CAC, and then a process called electron transport. All that stuff happens little by little. And just as an overview, one more time, I'm going to show you this again. Let me erase this stuff here. Let's see. I'm going to color code those 12 hydrogens. Right there. Now remember, little by little and through the process of glycolysis and the citric acid cycle and all that stuff that I just mentioned, these hydrogen atoms get ripped out of glucose and become a part of NAD, and NAD plus becomes NADH, and FAD becomes FADH2. So all of these hydrogen atoms get ripped out of glucose by NAD plus and FAD, and they become NADH and FADH2. These guys bring those hydrogen atoms from glucose into the mitochondria. So we get NADH and FADH. Oops, I'm not color coding properly here. H2. And there the hydrogens are used to make ATP. Hydrogens used to make ATP. When they're done with that, those hydrogens join with join with O2 to make water. So it becomes H2O. After making a bunch of ATP. So let's look at how that is done. The first way that this is done, first process is, let's not do red, let's do blue, is the process of glycolysis. All right, so glycolysis, first up, it happens in the cytoplasm of the cell. So we're not in the mitochondria yet. happens in the cytoplasm of the cell. It begins with glucose. Okay, which is a 6-carbon molecule. And it ends with a molecule called pyruvate, or let's say 2 pyruvates. All right, let's look at the details. Now, you've seen me write all of our chemical reactions, horizontally, going this way, like substance A plus B become product C plus D, that sort of thing. Not doing that this time because we're going to run out of room. I'm going to do things vertically. Alrighty? So, C6, H12, O6. Let's keep color coding here. All right, that is a 6-carbon molecule, and I want us to keep track of those carbons, all right? So what if I draw the carbons like a little ball? 4, 5, 6. Like that's glucose right there. All right, just to kind of keep an eye on that, on what's happening to glucose in general. All right, it undergoes a chemical reaction that you don't have to memorize. I promise you don't have to memorize any of these. I mean, heck, you're doing an open note test anyway. But a chemical arrangement occurs, and it, changes things up just a little bit. Something like that. But there's something, you know what, I'm going to make this arrow a little bit bigger here. There we go. Something funny happens with this chemical reaction. And I will show you something funny right there. ATP gets used up. ATP becomes ADP. And actually, if you're curious, it puts one of the little phosphate groups on there. You don't have to know it, but know that. But what you do need to know is that this first chemical reaction uses ATP. ATP, now let me write it over here. ATP losing that initial phosphate group and becoming ADP, is that endergonic or exergonic? Does that require the input of energy, or does that release energy? And dun dun dun, if you said, well, that releases energy, because we've written it like this before a gazillion times, you were correct. This is an exergonic reaction. It releases energy. But what is the cell using that energy for? You may ask. That is the burning question. It uses the energy released from ATP losing its phosphate group going to ADP. It uses that energy to power this one, the main reaction in blue, right there. So this one must be endergonic, like that. These are called coupled reactions. It's where an exergonic reaction powers an endergonic reaction. So here, this one, the one in green, ATP releases energy, that's exergonic. That powers this one in blue, this reaction that's represented by the blue arrow, which must be exergonic, because it needed ATP to make it go. Alrighty, and then there's another chemical reaction that occurs, and it rearranges things just a little bit, and I'm just going to draw like a little placeholder and a fill-in-the-blank line, because I don't care that you know what that molecule is. But this next step, there's yet another chemical arrangement, and fine, let's just go ahead and draw my six carbons. And you know what? Here, I'll put them here too. I don't want to be a lazy bum here. 2, 3, 4, 5, and 6. There we go. But on this one right here, another ATP gets used up, an exergonic reaction. So this is exergonic. Powers the one in blue, which must be endergonic. There we go. So I just want you to step back and take a look at this for a second. We started off up here with glucose. And it's undergone, what, 1, 2, 3 chemical reactions here, and 2 out of those 3 use up ATP. I mean, what the heck is going on there? I thought we were supposed to be getting energy out of this. Why are we using ATP when we're supposed to be making ATP? It goes back to that same old thing with enzyme, with the energy of activation in an enzyme, in that you have to spend a little energy to get more later. It's kind of like the lottery. You know, if you want to make money, you got to spend a little money first. So it takes a couple of ATP to really get this process going in the first place. The cell will make up for it and make more ATP later, but it does take a couple of ATP just to jumpstart this whole process of glycolysis. So let's go on. Down here, yet another chemical reaction happens. And we still have a 6-carbon molecule here. But then on the next reaction, really pretty cool, the next chemical reaction breaks this 6-carbon molecule in half. And we end up having 2 3-carbon molecules. 2 3-carbon molecules. So it basically just breaks this puppy right in half. Like that. Change that. So it's going to break that right in half. So now where we started off with 1 6-carbon molecule up here, it's now been broken in half into 2 3-carbon molecules. All right, now these 2 carbon molecules are going to be identical. This one over here is going to be identical to this one. So I'm going back to blue. Each one of these is going to undergo the same chemical reaction. All right, kind of like that. And what's interesting about this one is that this is the first time that that NAD plus thing is going to rip out a hydrogen right there. So let's do it on both sides, why not? NAD plus will become NADH. And again, those H's, they were somewhere on here. I don't know, I'll just put them in there for now. And now they're still 3-carbon molecules. So they're just missing a hydrogen. Then something pretty cool and interesting happens. Something a little different. ADP, oops, I can't write. ADP, 2 phosphate groups on there, gets its phosphate group back and becomes ATP. And it does over here. ADP becomes ATP. This is the opposite of what happened back here where ATP is used up. Down here, ADP gets its phosphate group back. And you know that that takes energy. So this is endergonic. This is endergonic in that it requires energy to put that phosphate group back onto ADP to make ATP. That means, well, where did it get that energy? That means that this main reaction in blue here, and here, must be exergonic. It must release energy. Exergonic. So the one in blue is exergonic, the one in green is endergonic in this case, and that's the opposite of what we saw back here. Yeah. Oh, so check this out. We used 2 ATP. We used up this ATP here and this ATP here, and now we just made them back, huh? So it's kind of like we spent $2 on a scratch ticket and we won $2. So now we're even Steven. Alrighty. So now we still got some 3-carbon molecules here. There we go. Oh, and just a reminder, you are not going to need to memorize each and every one of these chemical reactions here, but you do need to know how much NADH was made and how much ATP was made at each step. We talked about that as well. All right, so there are a couple more chemical reactions that occur here. You know, nothing too interesting going on. But one of the last ones here, this ADP thing happens again. ADP gets its phosphate group back and becomes ATP. Again, same thing we saw up here. So this is endergonic. We know that takes energy. So it must be this exergonic reaction that is in blue, releasing energy, and that energy goes into the ATP to form ADP to form ATP. And the same thing happens over here on the left. ADP has the energy to get its phosphate group back. That's endergonic. Takes energy. And that energy came from energy released from this exergonic reaction. Over here. Ooh. And this last molecule here is called pyruvate. Pyruvate. There, it's a 3-carbon molecule right there, and that is the end of glycolysis. That is the end of glycolysis. And it's happening in the cytoplasm. Yeah. Ooh, but let's summarize. Let's summarize here. Where can I summarize where that's not in the way of what I want to do here? So summary. I'm getting tired of these colors here. How about a nice plum color? Okay, so glycolysis. It used 2 ATP, but made 1, 2, 3, 4 ATP for a net gain of 2 ATP. Right? Now the cell also got some other goodies back here. It got these 2 NADHs. So you get 2 NADH molecules. Started with glucose, ended up with pyruvate. All right. In the cytoplasm is where this is happening. But we got to get into the mitochondria. So these two pyruvates get shuttled into the mitochondria. So this one goes to the mitochondria, this one goes to the mitochondria. All right? So we are now in the mitochondria for the next step, which is called the intermediate step, or sometimes it's called the transition step. So the transition step occurs in the mitochondria, and it begins with pyruvate. So it begins with pyruvate, and remember we have 2 of them. So I'll just keep drawing 2, and they are 3 carbon molecules. I don't know if you can hear the rain on my window right now, but it is pouring down rain while I'm I'm recording this for y'all. All right. But I'm nice and cozy in the house with a hot cup of tea. I hope you're cozy where you are too. Okay. The intermediate step, or transition step, begins with pyruvate, and we're inside the mitochondria right now. All right, this is some big old complicated chemical reaction. You don't have to memorize it, but you've got it in your notes. So any question that I ask you about it, you can just look this up in your notes. You'll have it there. All right. So I'm going to draw a big ol' arrow because we are seriously going to need the space for this. So what happens is that there is some weird molecule that actually helps enzymes do their job, and they're called coenzymes. And this one's called coenzyme A. So coenzyme A gets involved. Draw this here too. Only I'm not going to write the word coenzyme A again. I'm going to write co-A. Coenzyme A actually binds to two of the carbon molecules. And then what? And as it binds to two of those, to the pyruvate, it actually cuts one of them off. And that one carbon gets released as CO2. So I'll draw that over here too. Coenzyme A joins with pyruvate, and as it does so, it boots off one of those carbons as CO2. So we're only going to be left with two carbons on this molecule with that coenzyme A attached to it. But one more thing happens at the same time, because this isn't crazy enough. Let's go with a salmon color. NAD plus pulls out a hydrogen and becomes NAD, you guessed it, H. So NAD plus rips out one of the hydrogens and becomes NAD, wait for it, H. And that's it. It begins with pyruvate, which is a 1, 2, 3 carbon molecule, and it ends up with this coenzyme A attached to two of them, and this is called acetyl-CoA. That is its name. Acetyl-CoA. Again, you don't have to memorize that. You just need to be able to find it in your notes. And I know this is on that worksheet homework that you have. That's also due on the 23rd. You got an email about that. So that's also due on the 23rd. And I'm going to ask you some of those questions on the exam, guaranteed. So do your homework. It'll make your exam go a lot faster, a lot easier. All right. So that's it for the transition step. One of the carbons is booted out as CO2. So we've made two of those CO2 so far. Now that hasn't happened before, has it? What if we roll back up here to our C6H12O6 thing right here, where we're producing CO2? Let's use those little tick marks, you know, like you go like this for stuff. Let's put two dashes there. That we've made two of those, haven't we? Now let's skim down, get back up here. The transition step, intermediate step, it happens in the mitochondria. Coenzyme A gets involved. You lose two CO2s. It ends, we made two NADHs, and we ended up with two acetyl-CoAs, CoA molecules. That is the intermediate step. Dun, dun, dun. I'm just taking you through the process so that you can see what's happening to glucose. You don't have to memorize this. You just need to have things in your notes and be good with your homework, know what's going on. All right. So, what is next? What is next is the citric acid cycle, or the CAC. I'm going to abbreviate that. CAC. It's a cycle because the beginning product is also kind of sort of the end product here. And but as we go through this, you're going to see what I mean here. All right, so the citric acid cycle. I'm only going to draw for one over here. Okay, I need a place for notes over on the side here. So the citric acid cycle, it occurs in the mitochondria. That's where we are right now. So it occurs in the mitochondria. It begins with acetyl-CoA, just like that. And the first chemical reaction that happens with this acetyl-CoA, right here, it undergoes a chemical reaction, and hanging out in the cell, it is a molecule called oxaloacetate. I can't believe I even remember that. Good grief, you don't have to know that, oxaloacetate. But I do happen to know that it's a 1, 2, 3, 4 carbon molecule, and this 4 carbon molecule combines with the acetyl-CoA here, becomes a 1, 2, 3, 4, 5, 6 carbon molecule. Wait, I messed something up here. 1, 2, 3, 4. No, it is. It's a 4 carbon molecule. Yep. So we end up with a 1, 2, 3, 4, 5. There we go. 6 carbon molecule that it becomes. And as those get together, acetyl-CoA, coenzyme A leaves the building. It's done. And the first molecule that is formed is called citric acid. Spoiler alert, it's called citric acid. It's called the citric acid cycle because the first molecule that is made is called citric acid. We used to call this thing the Krebs cycle. Sometimes it's called the Krebs cycle. Probably because somebody whose last name was Krebs discovered this and spent their life's work studying this. And if you discover something in science, you usually get to name it after yourself. But that doesn't tell us anything about this metabolic pathway. Like I could call it the Snooki cycle, and that would tell us just as much. Right? So we call it the citric acid cycle because, citric acid is the first molecule that is made right there. So, all right, it undergoes a chemical reaction, several more chemical reactions. And in these chemical reactions, 1, 2, 3, 4, 5, 6, NAD plus pulls out another hydrogen and becomes, wait for it, NADH again. And then another chemical reaction occurs. Oop, I did not leave myself enough room there. NAD plus pulls out yet another hydrogen and becomes NADH. And this thing is still going to be 1, 2, 3, 4, 5, 6 carbons long. I need some more room here. Then one of these carbons gets chopped off and exits the building as CO2. So this thing's going to be like a 1, 2, 3, 4, 5. I'm making my molecules smaller now to fit this all on the page. It becomes a 5 carbon molecule. Then another funky thing happens, and this 1, 2, 3, 4, 5 carbon molecule, this chemical reaction releases some energy so that ADP gets its phosphate group back and becomes a ATP. There we go. back to blue here. And then, ooh, one we haven't seen in a while. One we haven't seen yet, anyway. Let's do something in orange here. This FAD thing rips out 2 hydrogens and becomes FADH. Back to blue. 1, 2, 3, 4, 5. And in the next chemical reaction, one of these carbons gets chopped off again and is released as CO2. And so I'm going to show a CO2 molecule coming out of there. There's one there. 1, 2, 3. So we're left with just 4 carbons now. And then another chemical reaction occurs, and this whole molecule becomes oxaloacetate again. And so this whole thing feeds back into itself. Can't get this all on one page, but meanwhile that NAD plus pulls out one last hydrogen out of there. So this thing is a cycle. One of the starting molecules, oxaloacetate, is the same as the ending one, so get rid of that. And so all this stuff that happens to this acetyl-CoA happens to this one. So let's add up the goodies that we get from just one turn of this cycle. We got some goodies here. We got some NADH. 1, 2, 3. We got 3 NADH. I'm getting tired of color coding here. Right, we spit out 1, 2 CO2 molecules. Oops, FADH2. We got 1 FADH2. That's yummy for the cell. And we did get an ATP out of this, didn't we? So check that out. 1 ATP. But we need to double all these numbers, don't we? Because this acetyl-CoA went through the same thing too, right? Or to summarize, let's back this up just a little bit here. Let's go ahead. I'm just gonna, no, maybe I won't. H12. C6, H12O6. Remembering glycolysis, it underwent a whole bunch of chemical reactions that finally split this thing into 2 3-carbon molecules, right? And we eventually ended up with 2 pyruvates. And this process here was glycolysis. And you already have in your notes that it happens in the cytoplasm of the cell, but it used 2 ATP, made 4 ATP for a net 2 ATP. But we also got 2 NADH out of that, didn't we? And then these 2 pyruvates here underwent that crazy coenzyme A transition step thing. An NAD plus became an NADH, and we ended up with acetyl-CoA. So the transition step, this happens twice. I can't spell. Acetyl-CoA. So we got NADH, we booted out a CO2, we lost 2 CO2, made 2 NADH, and then each one of these acetyl-CoAs undergoes the citric acid cycle that we just went through, and each one gave us 3 NADHs. What the heck, I'll color code there. 1 FADH2, 2 CO2, and 1 ATP. All right, so 3 NADH, 1 FADH2, 2 CO2s, 1 ATP. So for the citric acid cycle, and again, we're in the mitochondria here, we got to add these two together, don't we? So we got a total of 6 NADH, a total of 2 FADH2s, produced 4 CO2s, and we got a total of 2 ATP there. Oh, by the way, these CO2s here, 4 CO2s, let's go way back here and add our little tick marks to the other 2 that we had. So let's see, 4 CO2. So we already had 1, 2, so and we're adding 4. 1, 2, 3, 4. That is 6 CO2. I love it when the math works out. Isn't that fabulous when it does that? Love it when the math works out. So yay, this whole part here is done. That is done by the end of the citric acid cycle. And so far we have not seen O2 get involved at all. Remember, that happens last. Last. Okay, so, but back here. All right. Back here, we started in glycolysis with C6H12O6. We got 2 ATP out of that. You know, we used 2, but we made 4. We got 2 NADHs. They underwent the transition step inside the mitochondria, in mito. And we ended up with 2 acetyl-CoAs. Ooh, I left my L out of there. There we go. And each one of those undergoes the citric acid cycle, like that. And we got all kinds of goodies out of these. This whole part here now, C6, H12O6 plus 6O2 becomes 6CO2 plus 6H2O. All right, remember how we connected these things? Oh, I'll use a different color. We connected these here. And I was like, we're like, okay, glucose here is losing hydrogen, so that's an oxidation. It happens first in glycolysis, transitions reaction, and the citric acid cycle. That part is done. And remember, all of these hydrogens got picked up and became picked up by NAD plus and FAD and became NADH and FADH2. Maybe I'll color code that too, just so you can see. Oops. There we go. Those hydrogens here came from glucose. This is where we're at right now. None of this stuff here has happened yet. All right. But before we talk about this part here, let's add up the goodies that we have so far from glycolysis and the transition step and the citric acid cycle. So let's make a little table for ourselves. Let's come down here and let's glycolysis, the transition or intermediate reaction, and the citric acid cycle. And let's figure out how much ATP we got from each, how much NADH we got from each, and how much FADH2 we got from each. So for glycolysis, did we get any ATP? Go take a look. Well, we used 2, but we made 4, so we got a total of 2 ATP from that. Did we get any ATP from the transition intermediate step? Go back and look. Here's the transition step. Oh, I don't see any ATP there. Nope. No ATP. Citric acid cycle, did we get any ATP? Oh, yes, we did. There's one. Ooh, and there's another one, so total of 2. So we got 2 ATP from that. All right, how much NADH did we get from glycolysis? If you said 2 NADH, ding, 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 you're a winner. Let me write this down right here. There we go. Did we get any FADH2 from glycolysis? Nope. How about any NADH from the transition or intermediate step? Ooh, we did make 2 right there. Yeah. So we got 2 NADH. We did not get any FADH2 there. Citric acid cycle, how much NADH did we get? Don't say 3, because we got 3 for each turn of it. 3 times in that one, 3 times in this one, because we had 2 acetyl-CoAs from our 2 pyruvates. So a total of 6 NADH and a total of 2 FADH2s. So 6 NADH and 2 FADH2s. Let's add this all up, shall we? Grand total of 2 plus 2 is 4 ATP. 2 plus 2 plus 6 is 10 NADH. And 2 FADH2s. Check this out here. 4 ATPs. The cell just went through all this BS back here, starting at the beginning of glycolysis, going through all these machinations and biochemical gymnastics and stuff that is making everybody's head spins for a total of 4 ATPs. Yeah, the cell just went through all that, and all it got out of this was 4 ATPs. My parents went to Disneyland, and all they came home with, and all I got was this crummy 4 ATP t-shirt. That's nothing. That's nothing. The cell did not go through all of that just to get 4 ATPs. And here's why. Because these are the goodies. These are the goodies. The cell loves them. They love having all these NADHs and all these FADH2s. And remember that those hydrogens come from glucose. They were in glucose a long time ago when you ate something with sugar in it, with carbs in it. Those hydrogens are ripped out of glucose. And now we've got 10 of these loaded up with hydrogen and 2 of these loaded up with hydrogen. The cell is going to use these in the mitochondria to make a crap ton of ATP. Way the heck more than 4. Way more. Okay, the cell will use these. Let's write this down. Cell will use these to make a crap ton of ATP, way more than 4, in the mitochondria through a process called electron transport. Electron transport. There we are. Before we can go into the details of electron transport, we got to talk a little bit about the mitochondria and what's going on in there. I really want a really good orange here for the mitochondria. That's a better orange, more of a tangerine orange for the mitochondria. All right, so here is the mitochondria right there with its outer membrane and its folded inner membrane called the cristae, whatever. There we go. Remember, these folds are called the cristae. Okay. Inside the mitochondria, inside the inner membrane of the mitochondria, there are a whole bunch of, let's make them purple, why not? Of proteins that are stuck in the inner membrane of the mitochondria. And I'm just kind of drawing them in purple. These are proteins. They're really more like protein complexes, like this one right here that I'm drawing off on the left here, this one here. That's made up of tons of different proteins. This one here is made up of a bunch of different proteins. So really they're protein complexes that are stuck in the inner membrane of the mitochondria, and they are called the electron transport system, or the ETS. The electron transport system is a series of membrane-embedded a series of proteins. I'm going to say a network of proteins that are stuck in the inner membrane of the mitochondria. So let's write this down. It's a network of proteins stuck in the inner membrane of the mitochondria. Oops. Word those together. The mitochondria. Okay. Some of these proteins let's say they play with electrons, whereas some of the other proteins and some of the proteins, let's say they play with protons. And you know this thing is a proton from watching the last lecture, last video, a previous video, not our last, that's for sure. You know these things are the same thing. All right. So what they do is that they take the hydrogens that came from glucose that are brought there by NADH and FADH2. So let me put NADH back up there. Oh, no, yellow. All right, I'm just going to stick with NADH here. All right, I'm not going to draw all 10 of them. I'm not going to draw FAD as well. Let's not do that just yet. But remember that NADH got this hydrogen from glucose, from C6H12O6. All right? And they bring them to the mitochondria. Once NADH accumulates inside the mitochondria, it's like, you know what? Here you go, proteins of the electron transport system. I'm going to go back to being NAD+, and I'm going to give up those hydrogen atoms to you in the form of 2 electrons and a proton. And what happens is that, well, in the electron transport system, some of these proteins are like, oh, hey, I like to play with electrons. Oops, did not mean to do that. Erase that. Okay, let's say this protein complex takes those 2 electrons there. Whereas the protons get pumped across that inner membrane of the mitochondria. Like that. Let's do this again, only I'm going to draw it a little bit bigger. because this mitochondria here is taking up more space than I can let it have, because we need a little more space than that. I'm just going to blow up and draw this part here. So let's come down here, and I'll draw, what am I doing, the mitochondria? So yeah, the tangerine. Let's do, there's the outer membrane, and here's the inner membrane. All right, let me put all these protein complexes of the electron transport system back in here. E4, this one here. Oh, and by the way, this is kind of repeated all throughout the entire mitochondria. Like, for example, back here, they're just this whole thing is just going to have a ton of these. They're just going to be everywhere. That's that one that I drew like a funnel or pilgrim hat. There we go. So like that, but that's getting messy. Okay, so I'm just going to draw one here. Okay, so these are, in purple, are proteins of the electron transport system, so ETS. Some of these proteins like to play with electrons, and some of them like to play with protons, and you'll see what I mean in a sec. And all these NADHs yellow, are like, okay, now I've ripped all these hydrogens I've ripped a hydrogen out of glucose. Here you go. I'm going to give it back to you, but in the form of 2 electrons and a proton. And I'm going to go back to being NAD+, right there. These electrons get picked up here, and let's say that the proteins of this protein complex here kind of play hot potato with those electrons. They just kind of bounce them around from protein to protein. Meanwhile, so while these electrons are playing a game of hot potato with some proteins there, these protons the proton gets pumped across the inner membrane of the mitochondria. Now think about where this proton is right now. I mean, what would you call that space? This is the, matrix of the mitochondria right here. And way out here is the cytoplasm. It's not out this isn't in the cytoplasm. It's in this space between the inner membrane and the outer membrane. It's really, the intermembrane space, which I believe is one of the answers on your homework assignment. So, there's that. Great. But think back to these NADHs. Didn't we add them up and decide that there were, 10 of them? Yes, 10 of them. So let's put a 10 in front of this and say, okay, that means that not just 1 proton, but 10 protons are going to be pumped across the inner membrane. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right. We have a situation here where we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, a concentration of 10 protons in this intermembrane space, but on the other side of the membrane, we have no protons at all. Folks, what is that thing called when you have a high concentration in one area and right next to it have a low concentration of that stuff right next to it? If you said concentration gradient, you are the winner. Ding, 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 ding. This is a concentration gradient. Is a concentration gradient stable? No, a concentration gradient is not stable. This is not a freaking stable situation here. Nature does not like 10 up here and 0 out here. So what's got to happen? You good with the idea that 5 of these need to get out of here? 5 of them need to move from here down in here? Yeah. Back about a week and a half ago, when we were talking about concentration gradients with diffusion and osmosis, and we talked all about how concentration gradients aren't stable and nature wants them to be equalized, I told you in your notes to circle a super important sentence and put stars and bells and whistles and swirlies, whatever you could think of to drag your attention to that, to remember that sentence. Can anybody think of what that sentence is, what that concept is? And it has to do with the concentration gradient. So if you want to go back and look, here's where if we were in person, I would take a sip of my coffee and wait about 3 or 4 minutes for somebody to find that sentence. And right now somebody's finding it and they're saying, when a concentration gradient is equalized, energy is released. You would be very correct. Energy is released when a concentration gradient is equalized. Yes, I put an equal sign instead of writing the word equal there, equalized. That is when you put little gold stars and swirlies or whatever you needed all around there. Okay? Energy is released. Remember that, because we're going to equalize this gradient now. Okay, so we got a freaking concentration gradient here, and it is incredibly unstable. So, if we've got a concentration of 10 in here and 0 inside the mitochondria, 5 of these need to osmose through, right? Osmosis is diffusion of water across the semi-permeable membrane. These protons are a component of water, but it's diffusion across the semi-permeable membrane. They are going to diffuse through, but they are not going to diffuse through over here or through here or anything like that. Not happening. They're going to diffuse through this one protein of the electron transport system that I drew to look kind of like a pilgrim hat. Oh, it doesn't really look like a pilgrim hat, does it? Here we go. Let's make it look more like a pilgrim hat. Makes me happy to do that. Alrighty. Where's my yellow? There we go. They're going to diffuse right through this protein here. This protein here is an enzyme. It has a name, and it's the only one whose name that I want you to know. It is called ATP synthase. It's an enzyme. It ends in ACE, and we try to name it after the reaction that it actually works on. What does it sound like the enzyme ATP synthase works on? What kind of reaction would you expect ATP synthase to work on? Well, synthesize means to build something. So if it's ATP synthase, maybe it builds ATP. And you would be right. It builds ATP from ADP. Let's take a look at that chemical reaction there. I'm going to get rid of this here. Get these specks out of the way. You know that ADP becoming ATP, yeah, and what the heck, let's just draw the little phosphates there. 1, 2, 3. It requires energy. Energy's got to go into it. But it also requires an enzyme that I'm calling, I'll leave it in purple, ATP synthase is required to do that. But it takes energy to do that. What the heck? Where does ATP synthase get the energy to do that? From the flow of the protons through it to equalize the gradient. As these protons, and how many need to come through? 5, did we say? 1, 2, 3, 4, 5 need to come through, so I can erase 1, 2, 3, 4. Now let's erase this one over here. 5. Nope, I erased 1 too many. Whatever. Put 1 back. So 5 of those went through here. That released energy because that gradient was so unstable. This stabilizes it, and as it stabilizes the gradient by diffusing back through ATP synthase, energy is released. ATP synthase uses the energy released from the protons flowing through it to equalize that gradient to put that phosphate group back onto ADP to form ATP. And the cell goes, yay, we just made a ton of ATP to do that. ATP. There we go. All right, that is a lot to digest, isn't it? So I want you to go through this again, watch this part of the video again, and for next week's Tuesday lecture, remember we're not having a test on Tuesday, we're having the exam on Thursday, so I will talk about this again. I will take you through this whole diffusion process here one more time, and you'll have a little more time to let it sink in. This whole process, FYI, of these protons setting up an unstable concentration gradient that releases energy when they equalize the gradient through ATP synthase, and ATP synthase uses that energy to pop that phosphate group back onto ADP to form ATP. This whole thing is called chemiosmosis. And that, that actually is on your homework, that word chemiosmosis. So take a look at that. You can work on some of your homework now. And on Tuesday, I will recap all of this, and we'll finish this up, and then we'll have an exam on it on Thursday. Again, the exam is on Thursday, April 23rd. It's open note, it's open book, so you don't have to memorize this stuff. You just need to have taken the time to take good notes and to know where it is. You will have 75 minutes for it, unless you have an IEP, in which case you'll get double time. And I bid you adieu. Well, au revoir. Until we meet again, which will be next week. Take it easy and stay safe and stay healthy.